that is basically trying to use that biology to expand the pool of beta cells in an individual's pancreas who has type 1 diabetes. And so we're taking a number of approaches, from differentiating progenitor cells, from trying to redifferentiate cells from other cells. That's called transdifferentiation. It turns out in a mouse, we haven't been able to show it in a human yet, but you can take alpha cells in the pancreas and turn them into beta cells. And we're looking at trying to proliferate the small number of cells that an individual may have, so that we could uncover some biological function of those existing beta cells. If we can damp down the immune response at the same time, we can have some biological control, which we think will have clinical significance. And then we also have beta cell survival program going on, and are there agents, many of the type 2 diabetes agents may actually have potential to be therapeutic in a type 1 setting. Again, something surprising that if we didn't understand there was some beta cells in pancreases from people with type 1 diabetes, we would never have attempted those sorts of experiments. Now, the other thing we're starting to think about is the prevention of type 1 diabetes. This is a relatively new program at JDRF. We hadn't really been thinking about prevention in a significant way until the last few years. It turns out that type 1 diabetes incidence is increasing. It's really, at this point, you can see in a variety of countries, some epidemiology data, probably the longest data set from Finland. The incidence of type 1 diabetes is doubling every 20 years. So 3 to 4% annual increase. It's really striking when you look at data like this. It's not something we would have predicted. This is not about genetics, because the genetics is not changing in this time period. This is about something changing in the environment that's changing the incidence of type 1 diabetes. It's also changing the age of diagnosis, or the typical age of diagnosis of type 1. And you can see in this data, if you look at bands of age of incidence from 0 to 4 in the green, from 5 to 9 in the blue, and from 10 to 14 years of age in the purple, you can see that when one looks at the data, what the predictions are from what's been going on in Finland is that many more people are diagnosed younger. And so there's an increased incidence, and that increased incidence appears to be in that 0 to 4 age range. So that's telling us that we're starting to understand some of the incidence and genetic risk of type 1. And so if you look at sort of this graph, which shows the functional beta cell mass, or the number of functional beta cells one has in their pancreas, if you have a risk of type 1 diabetes, you probably have the normal 100% functional beta cell mass, and that's a genetic risk, and there's some environmental etiologies. And then at some point, something turns on the autoimmune response, and you start making autoantibodies. Some of you may have been volunteering for TrialNet, where we're supporting work to look at the incidence of the appearance of autoantibodies in siblings and close relations of people who have type 1 diabetes. But the first thing we can see that's sort of a signal that something's going on is this autoantibody response. After that, you start losing beta cell mass. And you already, in a sense, you have type 1 diabetes, but you don't have the diagnosis yet. It's not till that red bar where you've depleted about 80% of your beta cell mass that you actually get the diagnosis of type 1 diabetes. But the whole time that you have a falling mass of beta cells, you have type 1 diabetes. So as an immunologist, that's the time you want to start impacting the disease. And so we're very much trying to look for signals of how to identify individuals at risk so that we can impact them earlier. And so we think about prevention in two ways. Primary prevention, which would be, for example, universal childhood vaccination. It would be before the presence of autoantibodies. So there you would want to find a way to universally vaccinate everyone. Secondary prevention would be at the time you start seeing this autoantibody response. Is there something you can do to stop that autoantibody response? And so Our programs in this area in primary prevention have been on the viral vaccine front, where we're trying to understand the epidemiology of enteroviral infection and its impact on type 1 diabetes to see whether there's a potential for an enteroviral vaccine that could have benefit. You'll hear from Dr. Xavier later about microbiota, or the bacterial flora that you carry with you in a normal setting. There's probably some induced immunoregulation that may be related to disease incidence, and maybe by changing microbiota we can change the incidence of type 1 diabetes. And then we're also looking at ways, in the same way that people get allergy shots to turn off the immune response to a cat allergen or ragweed allergen, we're trying to ask, can we use the beta cell antigens as a vaccine or desensitizing agents to change the autoimmune response? Secondary prevention, we need to control the antibody response, the autoimmune response, and the beta cells. So we're looking at, again, these tolerogenic vaccines as well as beta cell survival agents. On the treat side of the house, we're trying to make the treatment of type 1 diabetes better, to keep people healthy so that they can benefit from these future therapies that we're trying to develop. This is data that comes from the T1D exchange. Those of you that don't know about the T1D exchange, you should look it up on t1dexchange.org. There's also a social network site called Glue for people with type 1 diabetes to interact with each other. T1D exchange has been starting to accumulate data from now over 60,000 individuals who are registered for the T1D exchange through their clinics, through their endocrinology clinics. And so they can accumulate data from these people with type 1 diabetes and see what's really happening out in sort of the regular endocrinology clinic. And this data shows you HbA1c's as a function of age of the person with type 1 diabetes. And as many of you in the room know, the treatment goal is to keep HbA1c less than 7%. And you can see from this data that out in the real world with 60,000, I guess this data, sorry, is from 25,000 individuals, we're not approaching the ideal of keeping HbA1c's less than 7%. And so what JDRF is trying to do is to find ways to make that possible and make it easier. So you know, for a very long time we've been supporting work toward the closed loop artificial pancreas systems. The goal there is to simulate a normal pancreas. There are many requirements to get a real closed loop system to work. It means improved glucose sensors. It means improved insulin that are faster acting, and other drugs such as glucagon. It means improved control software for the pump and the CGM. And it means the development of dual or multi-chamber pumps. And so JDRF has been supporting programs in all of those areas to try to really, in an incremental way, push the development toward what would be truly an artificial pancreas. We're starting to see first-generation approaches, either in clinical trials or in Europe already on the market. Many of you know about the low-glucose suspend pumps that are available in Europe, and we're hoping will be available soon in the United States. That's the first generation. Moving on, there's already clinical trials going on in the United States and in Europe on hypoglycemia minimizers. The next step would be have hypoglycemia and hyperglycemia minimizers. Those are all considered, at least in our view of the world, first generation artificial pancreases. Closing the loop between the insulin pump and the CGM really gets you to what we would consider a second generation approach. And again, JDRF's supporting work to really get to those second generation artificial pancreases. We're starting to see clinical trials in hospitals overnight. We're starting to see clinical trials of closed loops in camps for kids with type 1 diabetes, mostly at this point outside of the US, but I think this summer there will be some trials in the United States. And then that third generation where you really need multi-hormonal pumps and multiple hormones available to really approach substituting for the beta cell. A lot of progress being made here. This is an example that was published in February 2010. It was the first overnight controlled loop experiment in a hospital. Early successes really have demonstrated that using these sorts of systems, you can lower glucose variability, you can control to a very tight range, and you can reduce extreme hyper and hypoglycemia overnight. Obviously, we need to move that from the clinical trials testing centers to the home and then to really being available, but exciting progress being made in these systems. And I hope that gives you a sense of the excitement of what's going on at JDRF and the kind of work that we're supporting.